This is CheloniaCast, episode 59. Today we're joined by Dr. Blair Witherington, who is a research scientist and vice president for Inwater Research group based out of Florida that's doing a lot of really good work with sea turtles and marine life and sort of population analyses and holistically natural history work. He's also a member of the Marine Turtle Specialist Group, sort of this concentrated body of knowledge and task force of the IUCN that does a lot of good work with sea turtle and sort of all an area where all of this information can be concentrated. Dr. Witherington's written over 70 different articles, or written or co-authored over 70 different scientific articles. He's published a number of books. We'll have all of these linked in the description for those interested. Of those is including our Sea Turtles, a practical guide for the Atlantic and Gulf from Canada to Mexico. It's a gold medal award-winning book for nature. We're really excited to talk to him today about some of his research and Florida's sea turtles in general. So thanks for coming on, Dr. Witherington.
Well, I'm pleased to be here. It's an amazing podcast, and I'm sure you'll pose some very interesting questions. I love diving into the details of interesting questions.
Awesome. Jack, do you want to kick us off?
Yeah, I got you. So we kind of start all of these in the same way. What first got you interested in turtles, and what was the story there?
For anyone listening to the podcast, I don't have to describe how interesting turtles are. They're very approachable. They're very observable. They have a unique brand of charisma. So I've always liked turtles, but I've liked nature in general. And if I were to point to anything in my formative years that started me off, it would be my parents, of course. My father was an entomologist, and he encouraged me to kick over logs and see what was underneath. My mom encouraged me to get out, get dirty, and I did. So I was chest deep in local lakes, capturing all kinds of critters. And I decided I would be a biologist. And my principal focus was fishes, as a matter of fact. I was on the path to do a master's degree on fish, specifically a weed-eating fish, for a weed control type application. But I wasn't too excited about that. And right at that moment, I met a very influential person at the University of Central Florida, which is where I was going to school. His name was Doc Earhart. And he was a naturalist in the classic sense, in the sense of Frank Chapman or Archie Carr. In other words, he had a very broad interest in vertebrate animals in general and how they fit into nature and the world. And he himself was a very charismatic person. And I worked with him one summer on the sea turtle nesting beach here in Florida, and observed sea turtles nesting, and just realized that, wow, I'm trying to find a master's thesis topic, and these animals just have loads of unanswered questions. I have to work on them, and I really want to have Doc Earhart as my mentor. So I did, and it was all downhill from there. I worked on many different aspects of sea turtle biology, but became interested in marine life in general and beaches. And I'm still very interested in vertebrate animals, including reptiles, amphibians, and fishes.
So I listened to an interview that you did, or it was a memorial tribute to Dr. Carr, I believe, and you talked about a letter that you wrote in there, and that sort of set off your doctoral work at University of Florida, and that's sort of really a hub for sea turtle research. I'm curious what it was like to work in that sort of group of scientists and kind of early on.
Well, as I mentioned, Dr. Carr was a classic naturalist. So just being in his presence and chatting about anything at all was an amazing experience. He had obviously a wealth of life experience, but incredible insight on philosophical things about wildlife conservation and how we fit in with other species in our world. So He was an amazing person. So yes, when I was considering what school I might go to for a PhD, I really wanted to go to University of Florida. I wanted to work under Dr. Carr, and I wrote him a letter. I didn't think that he would ever respond, but he did, and that was his trademark. He was. I mentioned that turtles were very approachable animals. Well, Dr. Carr, like turtles, was very approachable. He would gladly talk to you about anything. And before I wrote the letter, I had met him at a conference and I presented at the conference and presented on a project that I was doing on hatchling production on a beach. And he had lots of questions for me. And so he at least knew a little bit about me, but he received letters from all over the world, from all kinds, because he was famous. He was Mr. Turtle. And anybody who wanted to work on sea turtles or turtles in general or vertebrates would love to work with Dr. Carr. So I was amazed that he returned my letter and wrote back a long letter giving me ideas as to what direction I might pursue and offering to introduce me to the faculty at University of Florida. And so, yes, he was instrumental in me ending up there, and I thank him greatly.
That's awesome. Thinking about just the amount of the letters he got and his influence, it's really just not only in sea turtles, but freshwater turtles too. I mean, the Handbook of Turtles was seminal, and it must have been reading about the tag returns he would get. I mean, that must have been so exciting to get a letter from someone abroad about sort of finding one of his turtles that he had marked. It just must have been incredible. And that
Oh yes, I mean, as you know, he dedicated a chapter in one of his books to that, and it's really funny reading.
Yeah. No, and he was a great sort of communicator broadly. Well, maybe we can sort of jump right into some of the research you did. I think sort of early on you focused a lot, and this has been a theme I think throughout your career and continues to be, but looking at the impacts of sort of photopollution on sea turtles, and that's a topic that it's interesting because a lot of the public knows about this because the public is intimately involved in this process and can mitigate it. But maybe you could start out just by giving us an overview of sort of what photopollution is and how it is unique in terms of a type of pollution.
Yes, well, when I first started working on this topic, it wasn't obvious to everyone that light could be or should be considered as a pollutant. So that's the most important concept there, that artificial lighting is a pollutant. That is, it's a hazard, it can cause harm. And thinking about controlling it or managing it gets us thinking about the way we think about other pollutants, like water pollution, air pollution, and so on. And so that's probably the most important concept that I would like for the general public to have in their mind. And that is that light, although we use it for our own purposes, we think of it as beneficial so we can find our way in the dark. It's also harmful and is a pollutant and causes harm to things that we care about in the environment. In fact, a very recent paper published in a law journal has looked at language in federal legislation for water pollution, namely the Clean Water Act. And so this law student has proposed that light pollution could even be considered and managed using the language that currently exists in the Clean Water Act. And so that's an interesting concept to think about. But how we regulate light and what we can all do about it from a public perspective and a private perspective really hinges on the emphasis on light as a pollutant, and that light that escapes our use and gets out into the environment can really cause a lot of harm. And it's also interesting that the light that escapes is not useful light. So in the subset of light that is useful, bouncing off a sidewalk so we don't trip at night, that light is not harmful to wildlife. It's the light that gets out, that bounces off of clouds, that reaches a nesting beach or a petrel nesting colony. That's the light that is harmful. And so you can think of it that way and believe that there are solutions at hand. Another important concept to understand is that unlike other pollutants, chemical pollutants, light is energy. And the moment we flip the switch, that pollution stops. So those concepts of separating useful and not useful, harmful light, and the idea that it's energy that can be immediately shut off really opens up a lot of solutions for us. So in theory, there's a lot we can do to remedy the problem, but of course in practice, it's complicated.
Right. It's interesting to think about it. It's a type of pollutant that, I mean, all pollutants can have these physiological impacts, but it's so evolutionarily linked with the cycles of different animals, and that it has this impact on sort of toggling behaviors and that sort of thing that maybe is more, it's kind of natural, but in a human sense, it's definitely sort of a bad anthropogenic issue. And so the thing about light that, as you've mentioned, it's energy, and it actually is a fairly complicated concept to wrap your head around if you think about it beyond sort of a surface level. There's a lot of different properties of light, and it's sort of a physics question to some extent. But what are the things and parameters about light that make it an issue, particularly for sea turtles? What are the things you have to think about with respect to light and how those physical quantities impact sea turtles?
Well, you're right. It is a physics question and a biology question, a behavior question, and there's also a very important human element, which I can get to maybe a little bit later. But from a physics perspective, well, I'm sorry. Let's start with the biology. So, from a biological perspective, we know that hatchling sea turtles pop up out of the sand at night. They emerge from their nest mostly at night, and then they're really good at finding the sea. When you look at the spread of a group of, say, 100 hatchlings coming from a sea turtle nest, on a nice dark beach, that spread is only about 25 degrees. I mean, that's pretty precise. So these are animals that have never seen the world. They are naive to everything. They pop out of the sand as a group, wipe the sand out of their eyes, and then go directly toward the ocean, which is where they need to be. So the question early on was, how do they do that? And for more than 100 years, we've known that they seem to use vision as their orientation, as the set of orientation cues that they use. So you blindfold hatchlings, they go around in circles. In fact, you blindfold one eye and they will turn toward the open eye. So there's a lot saying that the visual sense is important and that they balance visual senses between their eyes. But then there was light itself. So it was pretty easy to turn on an artificial light and lead hatchlings in the wrong direction. You could turn on a flashlight and just walk down the beach for hundreds and hundreds of meters, and the hatchlings would follow you. So it was clearly a visual sense. It was clearly light. That was the part of the world that the hatchlings were assessing. But you mentioned physics. What is light? It's energy, but it's complicated. There are attributes of wavelength, polarity, direction, shape, intensity, of course, and all of these things may play a role because, so we draw parallels to the way that we use light. We sense intensity. We know if it's really bright or not quite as bright. We sense color, so that's some ability to sense wavelength. But we can also sense direction. We know that this direction is brighter than that direction. But we also sense shape. We can actually see things. We can identify things. And so it's reasonable to assume that any or all of these might play a role in a hatchling recognizing the ocean. And as it turns out, those things do play a role. So wavelength, intensity, direction, and shape all seem to play a role. And this was really only realized late in the study of hatchling orientation, but it becomes a little bit complicated.
Right. Yeah, it's interesting. I just remember from sort of basic physics is it has a weird behavior that it can act as wave and particle, and that kind of gives light this interesting set of quantities that are unique to that. Um, and wavelength is one. The wave-like properties I think of light are kind of easy to analyze to a certain extent. And so you've looked at least for humans, a lot of time that's kind of how we measure uh, qualities of light and such. And you've looked at how different wavelengths of light sort of influence sea turtle behavior, and this is something well known. I mean, beachgoers know red lights, and it's something that's kind of accepted as you go on the beach with a red light. But what is it about the red light that makes it so that the sea turtles don't respond as much to that? And is there really no response, or what's going on there?
Yeah, that's. I guess I would say it's a pervasive myth that sea turtles can't see red light, and therefore any use of red light, no matter how bright it is or no matter how you use it, is okay. That's not the case. However, I often point to red light as a good solution for using light near a sea turtle nesting beach. Well, for a lot of reasons. The two key reasons are that sea turtles don't see it quite as well as we do. So in other words, we can see by red light better than sea turtles can. And that's based on electroretinography, how they are able to perceive light, and also their behavior, how they respond to light in terms of orientation direction. But also red light has a very special way it affects us. We can see by it using our cones, but with our scotopic night vision, which uses rods, that red light does not bleach out the rods that we depend on for night vision. So what that means in practicality is you find your way down a set of steps to the beach with red light, and then you turn it off and you still have night vision. By contrast, let's say you use a white light. You turn it on, have a white flashlight, you find your way down the steps, you turn off the white light, and you're blind. You're falling over, everything on the beach because you can't see anything. So that's one of the better reasons to use red light. But the most important part of the myth is that people on beaches with red light can do anything they want with impunity, and that's just not the case.
In one of your papers, you looked at the sort of response to different wavelengths of light, and it seemed like at kind of the tail end of the longest wavelengths, the red light, there was actually sort of a small rise in response, and it dipped down to the 650 nanometer range, or and a little bit before red. So is it preferable for yellow wavelength, or is there somewhere in between there that's an optimal? Does red light elicit a response, or how did we get to that point where that's the recommended?
Well, so you should know that part of the reason behind those experiments was to understand how we could change the spectral emission of artificial lighting that is like lighting our way near a dune, so that we could still find our way at night, but it would affect hatchlings minimally. I'm pretty sure, and most other researchers who have looked at this are pretty sure, that the longer wavelengths in the human visible spectrum, like from 600 nanometers down to 750 or so, are the best emission spectra for lights that are going to light our way on the dune, because of that intersection I told you about, with us still being able to see, but sea turtles not being able to see very well.
Right.
Having said that, when I did these experiments, one of the more surprising things I found was an extremely interesting behavioral response, not necessarily a perception change, but a behavioral response to light in about the 600 nanometer part of the spectrum. So that's amber light, deep yellow light. I used some sources that were monochromatic for that wavelength, namely low pressure sodium vapor. I also used special filters that only let through monochromatic yellow light. And I found that loggerhead hatchlings actually had an aversive response to that light, which threw me for a loop. It threw others for a loop. It seemed crazy. Light throughout the spectrum, even dim light, if you get it bright enough, causes hatchlings to move in the direction of the brighter light source. But in the yellow region of the spectrum, for some reason, that light caused hatchlings to go in the opposite direction. So loggerhead hatchlings. It was crazy. I published on it. I coined the term xanthophobia for aversion to yellow light. And I had every intention of following up on it. And I did, because I wanted to understand what the heck's going on. Why is this part of the visible spectrum different in terms of the behavior it elicits in hatchlings? I mean, how could it be functional? How did it evolve? Why did selection favor this behavior? And I came up with a lot of crazy stories and turned them into hypotheses and tested them, and none of them really panned out. So for any student that is listening and wants to follow up on what's going on with this strange loggerhead hatchling orientation behavior, have at it.
That's all. I mean, a lot of the listeners are younger, upcoming students, so it's always cool when there's sort of an idea that's tangible right there. That brings to mind something that's interesting about this that I hadn't considered this until recently is that you could have differences based on species in terms of response to lighting, and you notice that with loggerheads in particular, and sort of question why that is. Did you notice any other differences or grouped patterns according to species?
Other than the xanthophobia response that loggerhead hatchling showed, I worked with 4 species: hawksbills, olive ridleys, green turtles, and loggerheads. And they all had fairly similar responses to the spectrum, with the hatchlings favoring light in the shorter wavelength into the spectrum and longer wavelength light being less attractive. So they were pretty similar, really, which is astounding considering that these hatchlings are coming from beaches where females are nesting all over the world, from the tropics to temperate regions, rainy versus desert, all over the world. It's kind of interesting that this particular task of finding the seed from a nest involves, at least in the spectral aspects of the behavior, pretty similar cues.
Right. I'm curious in some of your, well, maybe sort of the evolution of this, but in one of your early papers, you used this V maze, and it was sort of experimental approach to looking at the impact of lighting on hatchling orientation and such. And that was pretty interesting. Maybe you could go into sort of what it was like to set that up and what kind of struggles were there. We spoke with Dr. Weineken on one of the previous episodes, and she spoke a bit about working on a similar project with magnetoreception. So I'm curious how just constructing that maze kind of went.
Yeah, well, I made it myself, like out of plywood in a shop at University of Florida, and the trick was to make something that was completely dark. So going back to what an experiment is. So I was interested in starting with the basic biology of hatchling orientation, understanding how hatchlings move where they move in an experimental sense, and then moving on to the field where things are more chaotic, but more real life. So these experiments with the V-maze, I intended to understand, the basic biology of the orientation. And in experiments, as you might know, the trick is to control all the elements in the experiment except for one. And in these experiments, it was the light and its qualities and intensity. So light is a difficult thing to keep away from an animal. You might think, oh, it's easy enough just to create a box where the only light stimulus is coming from this one direction. But it is not as easy as you think. Tiny little cracks in an experimental box can lead hatchlings in the wrong direction. And when I first started trying this, it was really frustrating because in my control treatment, the idea was there's no light. And I'm going to see if they choose alley A or alley B. Well, they chose alley B, and I'm trying to figure out why. And it's because there was a tiny LED on a charger off in the corner of the lab that was providing some dim light that got through the window, even though I thought I had blocked off every bit of light. So it shows you how difficult it is to create experiments where you isolate all the stimuli, and how much light there is in the world, and how sensitive the hatchlings are to tiny intensities of light. So, but eventually I cracked the problem. I made a lab as completely dark as I possibly could, which was not too bad to do here in Florida when I did the experiments, but I also took it on the road in Costa Rica, and I worked in an old generator building at Tortuguero. I worked really hard to board up the windows and make that thing as dark as I could, and it was a big challenge, but I did it.
So from thinking about that first attempt at sort of analyzing the question of how light sort of impacts sea turtle orientation and such, how have the metrics you've used to quantify that impact changed with time? And maybe not just what you're doing, but other people are doing, and sort of how has that changed?
Well, in terms of the basic biology, so we assume that a photon of light strikes the retina of the turtle, and the function of how a turtle's retina responds to light is a function of photon flux or photon intensity. So we can measure photons with spectroradiometers, and we can assign them to different wavelengths. But it gets a little bit tricky when you're talking about very low levels of light. So if you get on the internet and you search for spectroradiometer, you find all kinds of spectroradiometers that will measure light for you. That applies to photosynthesis, which occurs in daylight, or how we see the world as humans. Whether your computer screen is bright enough, but you look for ways to measure light at night, and even now, and certainly back then, the options were not abundant. So I selected a special photo multiplier tube that accepted input from a spectroradiometer that scanned through a grating. And not to bore you with the details, but the whole thing I had to assemble myself, and again, put in a big box that was powered by a generator that I had to take on the beach and in the lab and use it wherever I needed to use it. And even now, a lot of those capabilities are really only applicable for lab bench work. You can't just buy something off the shelf that will measure photon flux throughout the spectrum, something that you can take into the field. You're pretty much going to have to make it yourself. Although there's some really cool options involving what astronomers call a sky quality camera, which I could talk about later. It's an interesting piece of technology on the horizon. But to answer your question about how we measure light pollution from a management perspective, I would say that one of the more important elements in measuring light pollution are measuring it from a behavioral perspective. Like, go out there in the field and determine where hatchlings are going. Are they going in the right direction? If they aren't going in the right direction, which way are they going? Are they going north or south? Or are they dispersed 360 degrees? Those kinds of behavioral measurements are diagnostic for how bad a lighting problem is and what the lighting problem might be. And there are a few people tending to those sorts of data collection exercises now.
Right. So I think we focused a good deal on the light aspect of things and the fact that sea turtles are using that as a cue. I think a lot of times, though, with these sort of just figuring out what drives a biological response, like the urge that young hatchling sea turtles have to go to the ocean. I mean, there could be a sort of multi-variable, multifaceted dynamic going on. And so you've looked at this a bit with concepts like what else, this angle of acceptance concept and such, and what else is driving how hatchling sea turtles are orienting towards the ocean and how they know to sort of to go.
Yeah, so both the angle of acceptance concept and other aspects of their orientation, like how they respond to different wavelengths, go to the difference between what an animal perceives and how it responds to that stimuli. So for spectral properties, it could be that an animal sees the light, but it doesn't orient in that direction, like the whole xanthophobia response. Clearly, loggerhead hatchlings are seeing that light, but they're responding to it differently from shorter wavelength light. From the angle of acceptance, that is very different from a field of view. So imagine a turtle. Most turtles all turtles have eyes kind of on the side of their head, have a little bit of overlap of their visual field in the front, but they can see almost 360 degrees. In fact, the carapace probably prevents most of the vision directly behind them, but they have a pretty broad field of view. But within that field of view lies this angle of acceptance. So it's a special way that hatchling sea turtles, and maybe all turtles, are using light to identify a horizon. And so this angle of acceptance that seems to be confirmed from experimental evidence is a very broad angle horizontally, but very flat vertically. So the hatchlings can see overhead, but that overhead light doesn't count as much in terms of their orientation direction choices as compared to light that's near the horizon. And when you think about it, it makes sense because the key difference between the ocean and the land is horizon. The land has a dark silhouette from a dune or plants, and the ocean has a broad, bright horizon that you can see all the way down, including some reflection off of water. And so, like, going in the brightest direction, as specified by this very broad but flat angle of acceptance cone, you'd think that, yep, that's following that rule probably would get you in the right direction in most cases.
Right. That's really interesting and just demonstrates kind of the dynamic that it's not just the ambient sort of total amount of light. It's very specific and situation dependent, and sort of how that the light is being reflected and that sort of thing, and on that sort of horizon. It's something that you may have come across. I don't think this is necessarily 100% related to this topic, but I've looked a bit at coloration, scleral coloration in cooters, specifically Pseudomys turtles. And I'm just curious if you think, I mean, some of them will have very like light blues and turquoise sclera. And to me, that's always been kind of this, why is that? And maybe that's something that you've come across for eye coloration color and what that impacts.
Yeah, I mean, it's a fascinating topic because, I mean, for one, anytime you see variation in the natural world, you wonder, all right, why? Especially if it's constant, in a species. Like leatherback hatchlings have a turquoise sclera. And they're of course very different from pond turtles. So you might wonder, all right, what's going on there? I mean, marine turtles obviously live in a deeper body of water than pond turtles. Fresh water tends to be redder than seawater, which is much more blue. So in terms of spectral sensitivity, you might expect animals that live in deeper, clearer water to be more sensitive in the shorter wavelength blue into the spectrum. But the scleral reflection, that's light that you're rejecting. I mean, that's light that's shining out into the world that isn't getting specifically into your retina. And what adaptive significance that might have, I'm not sure. But it's an interesting point to follow.
Sort of an open question. Well, on this topic, I think that you mentioned this, the experimental side of things is good, but it doesn't capture the whole variation in nature. And uh, some of your work has looked at kind of in situ, um, how are the impacts of different stimuli, sort of like orientation and such, how are those sort of played out in a natural environment? And you've used tactics like actually tracking hatchling turtles with little flotations attached and that sort of thing. I mean, what is it like to actually do that sort of field work? That seems like it's kind of entertaining and enjoyable.
Uh, yeah, at the time, I didn't consider it entertaining and enjoyable. It was kind of arduous, but looking back, yeah, entertaining and enjoyable. I mean, those were the days. So you might be talking about some of the experiments where I put little floats on hatchlings, which emerge at night. So if you want to study where they go in the ocean, you're following them at night in a small boat, and it goes to a difference between like direct observation of animals and then inferential observation of animals, with inferential observation being maybe you infer where they probably go based on some remote sensing data or something. Well, these were direct observations. I mean, we saw where they went, we were there watching them. And it was a little bit of a trick to choose a tracking method that wouldn't affect their behavior. In fact, we used a little balsa float that had a reflector on it. So it didn't even have a battery or power source. All it had was a reflector. Pretty lightweight, pretty streamlined. And I think hatchlings swam at about 90 something percent of their swimming speed without a float. So we were happy about that. But the trick was hooking up the hatchling at night on a beach, launching an inflatable boat with an outboard motor, putting on night vision goggles, holding an infrared light, all on the bouncing sea, and following this hatchling for hours. And it sounds lots of fun, but it wasn't so fun at the time. So, especially for anyone prone to seasickness. So you're on the bouncing sea, and all you can see through the night vision goggles is a little green dot bouncing around chaotically in your visor. It was a good way to track the hatchling, also a good way to torture my lab assistants. But it worked, and we generated a lot of data. These were the days before GPS, by the way. We used a position indicating technology called Loran-C, which was not as straightforward as just hitting a button and recording waypoints on a GPS. So everything about it was really challenging, including where we worked, which was an Air Force base. So you imagine we're out there in the surf in an inflatable with night vision goggles in an Air Force base. All the security had a lot of things to say about, whether we should be there. Of course, we had permission, but there was always a new security guard that was questioning us. So everything about the work was challenging but memorable.
Yeah, right. I think it's important, yeah, to highlight that with I think there's the relaxing fun and there's the arduous fun, and the field work tends toward the latter, but it makes for good memories. It's like the concept of doing it and then remembering it in hindsight. You don't necessarily remember all of the pain and torture maybe that was attached with it, but it was a good experience overall.
You're exactly right. So anytime I go through that now, I realize, well, if I'm going to enjoy it later or laugh about it later, you might as well start laughing about it right now.
Right. Well, and okay, another thing too that with hatchlings and that's real interesting is this concept of, I don't really know where these numbers come from. We've talked to some people before on the podcast about this, but the 1 in 1,000 to 4,000 that make it. I imagine when you go out there and actually monitor hatchling turtles, you get a sense for how many are making it past that first 72 or so hours. I mean, is that 1 in 1,000 number accurate, or what have been your observations with hatchling mortality?
The 1 in 1,000 number, that's been around for a long time. Is it accurate? No, because it's a guess, and no guess is accurate. Is it close? Maybe. So it's based on population biology and lots of assumptions. So, for example, you can count how many eggs an adult female sea turtle lays. You can count up the number of nesting seasons she's likely to produce. And then you make an assumption about a stable population. And if the population is stable, then the number of female recruits is matching the number of females that are dying. And so you can take that number of eggs and say, all right, that's how many eggs it takes to make an adult female. A better guess these days is probably more around 500 or so, but it's still a guess. And it hinges on a good many assumptions, which vary in time and space. So it's like, is the population declining or is it increasing? And then in any given season, what's the mortality like during all of the life stages that sea turtles are going through? So it's a wild guess. It's okay, I suppose, as a framework to just let people know, oh, not very many hatchlings survive, and therefore survival of adults is extremely important. And in fact, that's probably one of the more important aspects of that number. For sea turtles and for turtles in general, or for any species that lives for a long time and produces offspring during its long reproductive life. Turtles, large fishes, that adult survival is critical. Hatchling survival is not as important, or that is, population trends are not as sensitive to hatchling survival as they are to adult survival. So if you're looking at threats to those larger life stages, adults, those are important threats that need to be addressed. And that's the lesson, really, I think, from that wild guess number.
Yeah, interesting. I mean, it's like with that sort of thing, life table analyses seem like they're fairly accurate, but with sea turtles, that sort of thing is very challenging to do. You need so much long-term data. It's almost, that's a huge undertaking.
Yeah, well, like for sea turtles, we have a whole lot of data at the earliest life stage, like eggs and hatchlings on the beach. And then we have some data for the latest life stages, which are adults. But there's 30 years between those life stages. And survival estimates in between are few and far between.
Right. With sort of going back to that, the tracking of the hatchlings, and there's probably certain experiences doing that kind of thing, that a harrowing experience, you could say. I mean, did you have any things that stand out that you were kind of in a situation where it was a little bit worrisome, or any crazy stories from that?
Well, sure, plenty. So a lot of the work involved being at sea in a small boat for long periods of time. And when you're at sea for days, things happen. And so these are days during which we're collecting data continuously. Like, we don't want to lose the turtle. We have a lot of investment in this turtle we've been tracking, and we don't want to lose it. Even if it's raining, even if it's dark, even if the wind blows up, even if we have a gale where it's horrific lightning and foamy seas, and it's dark, and the only hint you see of which way you're even pointing is when the lightning flashes, and you can see how big the waves are. And we just have to wait through those things and then find the turtle again after the storm ends. So a lot of those episodes seemed at the time to be horrific, but like you said, time eases out all of the bad memories, and you only remember the good ones. So I'm sure it was dangerous. Maybe we should have had more safety and backup systems. It seemed very horrific at the time, but hey, I survived. I'm here.
Yeah, I guess that's really what matters. Yeah,
Kind of taking things back to the xanthophobia. That's just wild to me. Like, what were some of your hypotheses? I mean, you said you never found any sort of answer, but like, why would loggerhead hatchlings avoid yellow light? That's just so strange to me.
Well, so as in a lot of science, you start with like storytelling to come up with hypotheses, and then you test them. So one of the stories I came up with was, when is orientation a challenge for hatchlings, and what are the spectral conditions like during those events? So I reasoned that maybe sunrise or sunset or moonrise or moonset might be a challenge because you have a really bright celestial source on the horizon. So it's the horizon that hatchlings seem to be measuring. And the horizon might not be, that sunrise or sunset might not be the direction that hatchlings want to go. It might be off to the right or off to the left or so on. During that period, though, the light is a little bit different. If you watched a sunset, you know that the light gets yellower and yellower, longer and longer. And the sun near the horizon looks rich with yellow. And in fact, that is the case when you look at it spectrally. A sunrise or a sunset, a moonrise or a moonset is richer in yellow light. So going back to this xanthophobia response, one of the interesting things I found was that I could take white light and make it brighter with yellow light and make that light less and less attractive. So everything was falling into line. This had to be the reason. This story had to be true. A source rich in yellow light that could distract or cause a hatchling to go in the wrong direction, like a sunset, could be dealt with. That orientation distraction could be dealt with by this xanthophobia response. It just seemed like too awesome a hypothesis not to be true. So I tested it using a lot of different filters around an orienting hatchling during sunrise and sunset. I tried to filter out the yellow light during a sunset on a south-facing beach where sunset was to the west. And I reasoned that, all right, hatchlings are going to have a hard time orienting when I filter the yellow light out of the sunset. But that wasn't the case. And I tried a number of different aspects, just making the light dimmer, filtering out red light, yellow light. And even though the hypothesis seemed an awesome one, it just didn't come out in the experiments. So again, for any student who's smarter and has some ideas as to how to test this hypothesis, I think it's something that's pretty rich, both from a basic biology perspective and from an applied biology perspective. Because the reasoning was that we can use this xanthophobia response to really lessen the effect of artificial lighting on hatchling sea turtles, at least loggerheads. And I think it still has both interesting implications from a basic biology and applied biology perspective.
Yeah, it's interesting to think about the evolution of it, as you mentioned earlier. Like, is this something that was just kind of a random mutation with loggerheads that has caused this, or could it really be something linked to a multitude of multivariable factors? That it could be something complex.
Yeah, and there's more craziness. I'll just mention briefly that a study was published on hatchling orientation in loggerheads from Australia using low pressure sodium light, monochromatic yellow light. Those Australian loggerhead hatchlings do not seem to have this xanthophobia response, uh, and that's crazy. So I can't explain why they have it, much less why one population has it and another doesn't, and it's just nuts.
That's
Yeah, it's one of those little niche things that's real interesting to sort of hone in on. Well, maybe we can move to sort of general Florida sea turtle sort of trends and more population focused things. But first, I think we'll just sort of start this out by just out of all your experiences and decades of experience on Florida beaches with sea turtles, what is the one experience that stands out the most in your mind?
Uh, all right, I've worked in some interesting places. I've had a chance to work on beaches all over the state and elsewhere outside the country, and in the water. Uh, and there are some interesting places. I'm getting ready to go down to the Keys. In fact, on Tuesday, and work on a long-term project in Florida Bay. It's been running for 30 years, and that project, as well as others, really teaches me that although it's fun to be in the field, you're out on the water, you're interacting with the animals, the wind is blowing through your hair. It's cool. The discovery often takes place, back at the lab when you're in front of a computer and you're looking at the data, and all of a sudden a spatial trend that you didn't know was there develops. I mean, that's for a pointy-headed science nerd. That's kind of the cool part of the science, is that something develops. I mean, that's the moment of discovery. It's often after you've run a statistical test and something that you didn't really know existed or you thought might exist, in fact, is significant. And it, I mean, for a nerd, it kind of overshadows the fun of being out in the field and being with the animals. But of course, being in the field is pretty cool too. And being in a place where the water is clear and you can actually like observe the animals behaving. You're catching animals that have a history. In this Florida Bay project, we regularly catch animals that have a decades long history with us. So we put in pit tags, passive integrated transponders that stay the life of the animal. And we'll, on a regular basis, have animals that we saw as juveniles and have matured into adults. And they have a record of 25 years or so, and it's amazing that they're long-lived animals. I mean, captures like that really drive that home, and it allows you that sort of peek at this very long life history that sea turtles go through.
Yeah, it's a really interesting point with sort of the analysis part of things. Like you don't really know what's going on until you sort of take it back to the computer or the lab. Something that seems to be consistent. I've followed this a little. I like the sort of data analytics side of things. Is a lot of sea turtle work in general, like in water things, require transect surveys, and that sort of you can estimate population parameters and sizes based on some assumptions about how many you're detecting and how many you're seeing and how that changes with distance from wherever you're observing and that sort of thing. And you can do that as sort of a one-dimensional thing, and then you can apply these density models to incorporate more of a three-dimensional aspect to this. I mean, how have you employed those tactics in your work, and is there, I guess, how have the analytics you're using for transect surveys evolved with time?
Also, the one of the basic elements of transect surveys that you mentioned was distance analysis, which is really important. So distance analysis, as you know, is a way to assign a detectability to animals, because you go out there and you look for turtles and you count 50 of them. Well, how many did you not see? Because that's how many are in the population. And so to understand how many you didn't see, you have to know what the probability is of detecting any individual animal out there. And you're right, they are distributed in space three-dimensionally, and some are available for observation and others are not. So one of the three-dimensional aspects of that availability comes from, in sea turtles anyway, comes from satellite telemetry, where you can put a satellite transmitter on an animal, you let it go, and then you find out that it spends, 3% of its time in the top, 1 meter of the water column, which is where you can see them on a transect survey, and it helps you, assign an availability to the detection function that you generate that allows you to estimate, density of turtles out there. Other than the satellite technology, that's pretty robust. Pretty robust set of methods that allows us to generate those densities. But I'll mention very quickly one thing I'm working on now as part of Inwater Research Group. We're trying to understand how many sea turtles are available for entrainment in this intake to a big power plant where we work. And to do that, we want to do line transect surveys in the area and understand how many turtles there are. Well, we weren't satisfied with our ability to see enough turtles at the surface. We are going to see them at the surface, and we are going to use that satellite telemetry to understand how available they are. But we're also going to use sonar, side scan sonar, to detect turtles in the water, to try to bump up our detectability of turtles in the water and fine-tune some better estimates of how many turtles are out there. And it's breaking new ground a little bit because the way you determine the detection function, eyeballing a turtle from a boat is a little bit different in terms of detecting it from a sonar unit. But we're pretty excited about what the possibilities are.
So, yeah, really interesting. And thinking about just what you can do beyond just estimating size, you can sort of link different habitat variables and kind of figure out density of turtles and how that's related to subsurface features of the ocean and surface features and that sort of thing. It's incredible. And with the evolution of analytical methods, it's sort of exponentially kind of increasing in what can be done. When it comes to sea turtle nesting in Florida in particular, there's a lot of there's so much going on that actually getting an estimate of or quantifying it is a challenging thing. And Florida's used this index beach concept for a while. I'm curious, does the index beach concept capture all the variability across the state, and how good do you think current estimates are of the total number of nesting turtles?
The short answer is yes, I think it does capture both spatial and temporal variability, and the estimates are pretty good, not perfect, not complete, but as wildlife population estimates go, I think they're quite good. In fact, I'd hold out sea turtles in the state of Florida as being one of the more robustly monitored wildlife populations. And much of that is because of the network of turtle people in the state of Florida, a whole lot of citizen science, so to speak, that contributes to these indices, and then management by the state agency. And now, because of that, we have, for a good many decades now, since 1989, really good indices of sea turtle nesting activity throughout the state of Florida, and that's 1,300 kilometers of nesting beach monitored every single year, almost during the entire nesting season. It's pretty astounding. There aren't very many other vertebrate animals that you can think of that are monitored that completely. And so in terms of like trusting the trends and even the general estimates of population size, I'd say those are pretty trustworthy data sets. The biggest limitation is that with sea turtles, we have these adult females that crawl up on the beach where they make marks in the sand and you can count the nest. That's cool. But this is the oldest life stage in the population and just one sex. So everything else going on with males, everything going on with juveniles that are maturing, which is including a developmental span of 30 years or so, we don't have a good estimate of what's going on. And so when we turn to the numbers, we trust them, but we realize that there's going to be a very worrisome lag between some horrible threat happening out there in the water to sea turtles and then what we see on nesting beaches. It's not a good position to be in from a conservation perspective.
Right. And that kind of brings up the question of, analyzing trends over time. With sea turtles, there's this natural variability that you have with remigration and that sort of thing. But then you have to try to figure out how much of annual variability in nesting is due to actual declines versus differences in where female turtles are going. So how is that? What is remigration something that is driving a lot of trends that are seen with maybe particularly loggerheads have been kind of a contentious issue in Florida, and how much of that is actually due to sort of mortality versus just differences in or differences in phenology, I guess, nesting?
Yeah, so that's obviously a very important question, and if you've probably seen the plot of loggerhead nesting numbers over the years. It's very bumpy. There's you can squint your eyes and see some trends, which are probably important. But if you're looking at like the span of just a few years, most of that noisiness in the nest counts is because of changes in remigration intervals. I mean, these are not real population changes. Sorry. But for the long-term trends, which is, say 10 years at a span, those probably are population changes. The trick is to pick out those population changes amidst the noise that's coming from changes in remigration intervals, where turtles just decide, hey, this is a good nesting year, production on the foraging grounds is good, we're fat and happy, we're going to migrate and nest this year. So in a lot of wildlife questions, picking out trends means picking the signal out from the noise. And the remigration interval noise is something that's really troublesome if you only have less than a decade of data. Thankfully, we've got multiple decades of data now, and so we can start to say meaningful things about population trends in the sea turtles that we've been monitoring in Florida. And there's some interesting differences between the species, and we think we know. We don't know why things are happening. We can speculate, but we think we know what is happening.
Right. The loggerheads is particularly interesting to me, and what's been going on with that, because it seemed like there was this from mid 1990s to early 2010 or so, there was kind of this shock that they were declining and this was a problem, and now it's kind of more stabilized and stochastic, like you said. Do you think their current conservation status reflects how they're actually doing in Florida?
I think so. If you mean the conservation status as, say, federal agencies determine it, I would say yes. one important thing to understand with loggerheads is that we aren't seeing a recovery. And to me, that's one of the more troubling things about loggerhead sea turtle conservation in Florida, because for a long time now, we've had the Endangered Species Act, we've had money thrown at conservation. I mean, we've been working hard to make more sea turtles. Of course, we can't make more sea turtles. Only sea turtles can make more sea turtles, but we're trying to kill fewer of them. And it's frustrating that all of that money and action hasn't resulted in bigger successes. And so a critic might say, well, sea turtles live a long time. Their population biology means, you should expect sluggish changes. Don't be too frustrated. But I compare the loggerhead nesting trend to the green turtle nesting trend. And maybe you have those two plots pictured in your mind, but the loggerhead nesting trend is kind of noisy, goes down a little bit, and then it's gone up a little bit, but not much. The green turtle nesting trend, keep in mind they're nesting on the same beaches, it's exponential. It bounces around a lot, yes, but when you fill in the gaps between the biennial pattern there, even with the noise, it is launching upwards. Green turtles are an amazing conservation success story. In my mind, green turtles are responding perfectly to all of our hopes wrapped up in the Endangered Species Act, where we're like really protecting an animal. We're not killing them as much anymore, and their populations are responding, and everything is cool with their conservation. You could say that we're working just as hard for loggerheads, and they just simply aren't responding. And to me, it points to what we're able to do in conservation and in differences between the life history of those two animals and where the threats are located. So where the intersections between the turtles and the threats are. And the short story is on the difference between loggerheads and green turtles is that loggerheads spend a longer time away from protected waters in the U.S., out in the mean streets of the wide open ocean, where they intersect really profoundly with some intense fisheries, namely longline fisheries for sharks and tunas and swordfish. So they just because of their life history, they have a much greater intersection with some very severe hazards. And even though we're working like heck to conserve them here in the U.S., there are a lot of really important threats going on in international waters that are hard to come to grips with.
Yeah, it's not a good reality, and definitely something that it's kind of a battle, right? And there's a lot at play there, and sort of figuring out how to make the best of both worlds for fisheries and the conservation of the turtles. With regard to the demographic gap you were talking about, that Dr. Carr speculated on a lot on these lost years. You've done some work looking at that and what juvenile turtles. I mean, once that first few years, sort of out in the ocean. I mean, what are they doing, and what have you observed with young turtles as they're growing?
Well, the way to start that story is the way that Dr. Carr described that span of sea turtle life history, and that is, the lost years. And so that implies that turtles are out there and they're kind of lost. Of course, they aren't lost. They're just lost to us. We don't know what they're doing. We know a little bit more about what they're doing now through both inference from remote sensing and direct observations of the animals out there in the open sea. I've done some of that. But it's still very much a mystery. Things like survival, very much a mystery. True predation rates and exactly how long they're spending in that open sea environment, still a bit of a mystery. But we do know something about what they eat and how they behave, how they interact with habitat out there. For example, The sea turtles here in the Western Atlantic, at least 4 species, loggerheads, green turtles, hawksbills, and Kemp's Ridley, all as small kind of hand-sized turtles, inhabit the surface of the open sea in frontal zones. Frontal zones that tend to press together floating stuff out there. Stuff including things that the turtles eat, weird animals that live out there at the surface of the open sea, and animals that live as part of the pelagic sargassum community, which is a very interesting community that lives out at the surface of the open sea. They aren't completely obliged to live within that community, and they don't just they do sit and wait and conserve energy and just nibble on what floats by, but they also swim away and find their own food outside of these areas. So they're kind of facultative in terms of their ability to sit and wait and conserve energy, but also swim and locate food. And when you think about it, they have to do that because this community that they live within is a nice place to live with lots of food, but it just all blows apart as soon as the wind blows, and it disperses and all that food goes away, and they have to find someplace new. So it's an extremely interesting, dynamic community out in the open sea that is interesting in itself, and it's cool that sea turtles are part of that.
And you've got an interesting sort of holistic ecology background, and can probably characterize these sargassum communities better than most in a lot of ways. So what are the young sea turtles actually eating in there, and also is sort of an addendum to that, how much of that is plastic and pollution? Because I imagine a lot accumulates.
Yes, it does, and more every year. So as part of the work we've done, so we go out in a boat, we find these lines of sargassum. So I mentioned that they're in frontal zones that tend to be linear. You can see them from afar. You target them, you go out, and so you might be 100 miles offshore, but you can work through these lines. You can see turtles, catch them with a dip net. They're about the size of a coconut, and you bring them aboard, and then you can ask them lots of questions. What'd you eat for lunch? How big are you? Do you have any wounds? And we start to learn a little bit about their ecology. We also record how close they are to a floating object and what the floating object is, what their behavior was when we first sighted them. So we learn a little bit about energy budget. But on the subject of what they're eating, we can give them a gastroesophageal lavage. So we squirt some water in their throat and see what washes out. And they're eating a whole wide range of animals that are out there in the sargassum community, including some tiny little things. And you might think that they're eating some things that are a little bit closer to what they would eventually eat as larger turtles in the shallow water benthic environment, things like crabs. But crabs, as it turns out, are pretty swift, and they're able to outrun a turtle. These turtles are focusing more on slower sessile things, like little polychaete worms that live within spiral tubes on the thalli of the sargassum. Hydroids, they're like 30-something species of little hydroids, which are basically naked corals that are living on the sargassum. They're also eating hydroid colonies, which include Porpita, which is a little blue button, Velella, which is the by-the-wind sailor. So these are freaky hydroid colonies that drift on the open sea and end up in these frontal zones. And you wouldn't think they'd have a whole lot of nutritional value, but sea turtles love them. So they're eating a lot of these easily captured things. But they're also eating plastics. Now, why are they eating plastics? Probably because this is storytelling, but they're programmed to latch on to anything that floats by. because they might not see it again. It's a big ocean, and this thing that floats by might be good to eat, or maybe it's not very nutritious, but it isn't going to hurt you. Those were the old days. Now, hatchlings are eating bits of plastic that have been broken up by ultraviolet light in the sea, plastics that form flat little shards that build up in the gut of a small turtle and perforate the gut, but also clog the gut, almost like blood platelet clog a wound in a blood vessel. And these plastics end up being really harmful. Of course, even a moderate amount of plastics ingestion, of course, dilutes the nutritional quality of the food you're trying to eat. But these hatchlings don't seem to be getting any feedback on that they've eaten something bad, and then they're going to recognize the thing and not eat it anymore. They keep on eating and eating and eating. And so turtles that wash ashore in storms are often packed with plastics. In fact, very close to 100%. This is a scary stat. Very close to 100% of small turtles that wash ashore in hurricanes, I mean, turtles that we would never see otherwise, have plastics in their gut. And a lot of them have so much plastic, it's the dominant item in their gut. So the question is this like a population level threat? How many turtles is it killing? Probably a lot, but the estimate is hard to come up with. Like what fraction of the population? Because these are the worst case animals that wash ashore in a hurricane. And then we have at the other end the plastics that we wash out of healthy turtles, and it's like 20% or so of the turtles have plastic, but we don't know how much it's harming them. These turtles are acquiring a plastic load during years of life in these frontal zones, and we really don't know how bad it is. We know it's bad, just don't know how bad.
I mean, some of the statistics of it's from stuff I've read in the pet, like 13 to 20% of dry mass is plastic in young turtles. I mean, that and what you said, 100% of stranded animals have it, and it's just that's a staggering thing to think about. And it for people that I think a lot of us don't necessarily need a utilitarian appeal to protect turtles, but for people that do, you think about how that impacts humans and kind of down the line and in other ways, and just the chemicals and toxins associated with it. It's that's a really scary thing.
Yeah, it is. And so one of the important aspects of plastics is they tend to adsorb nasty stuff in the environment, chlorinated hydrocarbons, other things that are toxic, and with high enough titers in the blood can cause all kinds of problems, probably causing problems for turtles because these adsorption particles out there in the sea have a really high surface area to volume ratio. So ingestion is a problem. But we eat stuff out of the sea too, and so plastics are everywhere. And it sounds like hyperbole that plastics are killing us, but in some way they are. Plastics really are everywhere. We're still putting them in the ocean in really high amounts. Like a whole enormous fraction of all the plastic that we make ends up in the ocean, and the sea breaks it up into smaller and smaller particles, and it becomes part of the food chain, and we're part of the food chain. And it's not just speculation. It's demonstrated that, if you eat a tuna, you're eating some plastic along with it. These are tiny little, nanoplastics, not just microplastics, but nanoplastics that have a very high surface area to volume ratio. They work their way through tissues. They get into fish, and we eat fish. And you might think, it's just plastic. Who cares? Well, over the long term, we live a long time. It could be important for us, and it drives home an important question. why should we care about turtles eating plastic? And that implies like a selfish question, like why should we care about anything? I mean, people who care about turtles and sea turtles or ocean life, they care about those things. But there are selfish reasons for individual people who might never see a turtle or care about them to want to do things about habitat in the wide open ocean, because we are part of that. Yes, be altruistic, appreciate diversity in nature, try to conserve charismatic animals like turtles, but realize that these things are connected to all of us. And even if you have nothing but selfish interests, you should understand that this kind of conservation, protecting these environments, are important for us. And we are definitely connected to the marine environment. Even in faraway places with strange sounding names out in the deep blue sea, we are fundamentally connected to those places.
Yeah, the concept of the ocean as sort of a connector and sort of mediator is a really interesting thing, and to think about the impacts and on humanity. Um, I think it makes sense to we focused a lot on hatchling turtles, juveniles, not as much on adults, and to kind of round out the discussion on ecology and life history. I think that makes sense, and I'm just curious where you think that, because in water, sea turtle research in the water is sort of a whole different animal from nesting surveys, that sort of thing. And you have experience with that. I mean, where in Florida in particular is there specific research need for understanding sea turtles in the water?
Uh, well, it is, of course, far more challenging to understand sea turtle biology and life history parameters like survivorship and threats to turtles in the water, because they're just harder to get at. Uh, but it's important because understanding the intersection between the animals and threats is an important part of management. So you can manage this threat and reduce mortality. Within Florida, one of the more important elements to consider from a sea turtle conservation perspective is boat strikes. A lot of people are familiar with manatees and boat strikes and the importance of that mortality factor. Well, sea turtles are also extremely exposed to and sensitive to boat strikes. Boat strikes are the most common identifiable source of mortality in turtles in Florida waters. We have a lot of boats in Florida, and they intersect with sea turtles off of nesting beaches and in inlets and on foraging grounds, and boats hit a lot of turtles and kill them. And they're killing an important life stage, adults and turtles just maturing into adulthood. These have a high reproductive value. That is, that they're high to the population. The population trends are very sensitive to mortality in these life stages, and so boat strikes are really important. But and so there's a really important human element in terms of managing that. What do? You have captains be vigilant. Maybe someday we might have some go slow areas in really sensitive areas, like right off of nesting beaches during the breeding season. It's a touchy topic, as anyone knows from the experience that we had with manatees and boats. People love to ride boats, and they don't want to be told what to do or how fast to go. So there's a touchy human behavioral element there. But maybe one of the most important things to understand about sea turtle conservation in Florida waters is that our sea turtles in Florida don't stay in our waters. They go to waters in other states. They go to international waters in the Gulf of Mexico. They go to international waters in the general northern Atlantic, and they go to other countries. And there in those waters, they intersect with a wide variety of threats, including extremely severe threats like gill netting and longline fisheries and trawling. And although Many of those fisheries aspects are regulated here in the U.S., and we're coming to grips with reducing mortality from those fisheries sources here in the U.S. In other places, especially in international waters where there are no policemen, there is a lot of fishing activity going on that captures a lot of sea turtles and kills them. Oh, sorry, I can't hear you for some reason.
You've muted yourself, Michael.
All right, we're good now. Yeah, and just on the concept of interconnectedness, it's interesting to think about from the conservation perspective of the sea turtles, and how it's sort of a global thing. Kind of an obvious thing with turtles in general, but definitely creates a lot of logistical hurdles in trying to implement protection, and probably really frustrating is. So and you mentioned this earlier. I've read a bit about this in the past, but some of the Key West turtle work that you do in the water. This is one of the few places, maybe two different places in the north, where you get hawksbill sea turtles, the juveniles, and the reefs there. I'm curious if you could expand a bit on what you've learned about the turtles there, and with emphasis on the hawksbills.
Well, our study area in the Keys, this is a study area that Inwater Research Group has been working in for a couple of decades now. Includes a variety of habitats in the Keys. So most of it is shallow water. Some of it is very shallow water. I mean, less than a meter deep. Seagrass meadows in places like the Marquesas Keys, even inside the Marquesas Keys, which is an atoll-like island structure with a lot of seagrass in between. But there's an area outside the Marquesas, which is extremely interesting green turtle habitat. I could talk about later. But so you asked about hawksbills. Hawksbills are famous for being coral reef animals. They have a lot of attributes that allow them to live on a coral reef. They have very thick shell scutes that limit the impacts from abrasion to wedging themselves underneath rocks. They have a long neck and a narrow beak that allows them to get in nooks and crannies and eat their favorite food item, which is sponges, which is crazy that an animal would specialize in eating an animal that is pretty toxic. And so they're specializing in sponges that are not, the nice soft bath sponges. They're specializing in sponges that have silica, spicules. As Annie Malin termed these sponges, hawksbills live on a diet of glass. Basically, almost like little glass shards that the animals are eating and are able to deal with. They're also eating some other reef animals like zoanthids, which are cnidarians that live in little colonies on the reef. So they're famous for being tied to coral reefs, but we also find them in seagrass habitat, and same places where we find small juvenile green turtles. So this is in water that's a meter deep, maybe a little bit deeper, and they're hunting through dense seagrass to find little sponges like chicken liver sponges that are hiding between the seagrass blades. So it's wrong sometimes to just fully characterize an animal as, hawksbills, they're just coral reef animals. They're not just coral reef animals. They also live in other habitats, including seagrass. But it's interesting that they capitalize on their ability to deal with and process a crazy food item like sponges that other animals just aren't able to eat.
Right. The mention of the green turtles, that population there, that would be something really interesting to hear about. And you've done some work with the foraging there, and I'm also curious, just as an aside to that, have you noticed any social behavior with the green turtles there?
Yes, and so that's something else that's crazy. So our group has studied some green turtles that are in a location called the quicksands. It's an area west of the Marquesas and also west of Key West, Florida. And when you go out there in a boat, you look at these waters. They're pretty shallow waters, clear water, and you see a lot of white sand. And you wonder, what in the world is a green turtle, which feeds on seagrass and algae? What are these animals doing here? And when you see a green turtle and another one and another one and another one, and they're all large animals, subadults and adult animals, weighing up to 500 pounds, and you see the abundance of these animals, you wonder what's going on. When you look closely underwater, though, you see seagrass. You don't readily see it from the surface because it's so densely grazed. Green turtles, as you might know, graze on seagrass, and they don't just eat it willy-nilly. They maintain grazing plots, and that is to say, they graze the seagrass and then they go back to it and graze it again. And they prefer areas where the lawn has been mowed to areas where the lawn is out of control, because the area with new growth gives them much more nutrition from the seagrass that they're eating. Well, it could very well be that green turtles not only benefit from their own grazing of a seagrass plot, they also benefit from their conspecifics feeding on that seagrass plot. And if you take that hypothesis to some of its limits, you might speculate that, well, then green turtles should hang out in a group, because a herd of green turtles will be more efficient at grazing, and they'll benefit from the nutritional aspects of the grazing. And also in general, it's kind of good for an animal to be in a herd, so to speak. Often behaviorists call this a selfish herd, where an animal isn't trying to benefit the other animals in the herd. It just benefits from the vigilance, where if one turtle startles and then swims away because of a big tiger shark, all the other turtles will swim away too. And there's also in safety in numbers and other benefits to be in a herd. Well, even though it made sense a little bit, it's kind of a crazy hypothesis because no one really sees turtles traveling in a herd. You just don't see that. So, but we thought maybe that's because you can't see turtles very well in the water. And we see an animal and then we see another one and another one. Do they see each other? Are they trying to stay around each other? We wanted to know if that were true. So we set up a whole grid of transects through this quicksands area, and we found that there was a cluster. We did a cluster analysis, found there was the cluster. Well, all right, maybe they're clustering on a resource. We found that the cluster would change with time, and it would move around. So we thought, well, they're not clustering on a resource. They're clustering with each other. They're herding. And we published a paper on herding in green turtles. And I still stand by it. It could be something that you only see in high density areas where there are some special benefits to be in a herd. So this quicksands area has a lot of exposure to the open sea. There are some large tiger sharks. We've seen a tiger shark eating an enormous green turtle, almost like corn on the cob. So it's a dangerous place for a green turtle. So they might benefit from the vigilance of a herd. They could also benefit from the grazing elements. A lot of that is storytelling, but they do seem to be spatially clustered and not always at the same place. So kind of cool, I think.
Yeah, that's very interesting. That and almost, I mean, that herd behavior, do you think that that's visual, or is there some other kind of sense that's keeping them together?
I don't know, and it's a pretty important open-ended hypothesis to test, because sea turtles do cluster in other situations. So you might be familiar with the Ridley sea turtles, Olive Ridley and Kemp's Ridley. They nest in giant assemblages called aribatas, where on a given night or day, there'll be no nesting. Next day, no nesting. Then the following day, you'll have 100 on the beach. The next day, you'll have 10,000 Ridleys all on the beach. So, offshore, they're staging that aribata, and you can see them all out there. So the question is, how are they doing that? How are they staying together? Is it visual? Is it pheromones? Are they emitting sound? I mean, certainly there is a social behavior because they're the elements of social behavior that we see. But how are they doing it? Still an open-ended question, and pretty cool and fruitful ground for a student who wants to study how turtles might be communicating with one another, because it's hard to escape the fact that they are communicating somehow using some kind of modality. Just how are they doing it? No one seems to know.
Right. Yeah. That's a fascinating thing. I spent some time at Oceanal last year, and unfortunately, I kept extending my flight to the point where I overstayed my visa
No.
in Costa Rica, but still didn't see it, and it happened about 3 weeks late from when it was anticipated. So it was just a really tough situation. But I thought about it a lot. So yeah. But yeah, maybe we can sort of wind things down with a few general questions, and as we sort of think about wrapping up. I'm curious for you in specifically, what are the trends related to Florida sea turtles, and maybe the threats facing Florida sea turtles that worry you the most, and what are the best ways to fix those?
Well, we talked a little bit about the trends in two different species, loggerheads and green turtles, and the interesting fact that the trends are different between those species, with loggerheads showing a lot of variability but no real recovery, and green turtles giving us a pretty satisfying exponential upward trend, telling us that for that species, we seem to be doing something right, or at least not so wrong. Those are the trends for those species, and I think those trends help point out threats in those two populations. The former threat for green turtles was that they tasted good, and we ate them, and we almost ate all of them. So the number of green turtle nests in Florida. In fact, Archie Carr published a paper in the 1950s when he discovered his very first green turtle nest in Florida. So he reported on 2 green turtle nests in Florida, which he felt was astounding. Since then, last year, green turtle nesting set a record. I think the number was 77,000 green turtle nests recorded on Florida beaches. So even if you account for difference in detection, how hard we're looking, I mean, it's an astounding increase in an animal population. And it traces almost precisely, in my mind, protection from the Endangered Species Act. So the thing that the Endangered Species Act does best is it sort of reins in our pernicious tendencies to eat everything. And so we were, catching a lot of green turtles and eating them. We were harvesting them commercially. So it stopped that. So the worst thing that we could do to an animal population, that is hunt them down and kill and eat every one, that stopped in the 1970s. And since then, the green turtle population has launched upwards. So for that species, that was an extremely important conservation remedy to the threat. Now, loggerheads are really different. Like I mentioned, it's bouncing around, no real recovery trend. You could say it's kind of stable, even though here in Florida, we don't kill them anymore. We aren't harvesting their eggs. We're doing at least something to protect them in our national waters. So it tells us that this threat might be not within the framework spatially that is protected by the Endangered Species Act. It's someplace else. It's in international waters. It's in other countries. And so it stands to reason that these threats are there. And in fact, we know of some key threats like longline fisheries that take tens of thousands of juvenile loggerheads every year, trawl fisheries that also take tens of thousands of loggerheads every year. We don't have a really firm estimate of those numbers, but we know the numbers are extremely high. So it all stands to reason that those threats that aren't completely addressed by the Endangered Species Act and international treaties, CITES, trade and endangered animals, fisheries regulations, a lot of fisheries treaties that span international waters, everything that we're doing to protect sea turtles and especially loggerheads isn't doing everything that it could out there.
Yeah, a lot to think about in that respect. And returning back to the issue of sort of lighting and thinking about in recent times, LEDs have sort of taken over the market, and there's a lot of this research and sort of increasing efficiency of lights. And it's my understanding that a lot of times increased efficiency comes at a cost in terms of producing shorter wavelengths that are worse for wildlife in general. And with that in mind, I mean, what are the biggest struggles with trying to implement, with the drastic increase in types of lighting and spectral quality compared to the past? I mean, what are the challenges in trying to implement conservation measures in that area?
Well, the most fundamental challenge is that superimposed on our efforts to try to manage light as best we can is human population growth and movement of people to the coast. People are still building on the coast, and when they build something, whether it's a condominium, hotel, house, they put lights on it. In Florida, that lighting comes under review. We try to, by we, I mean the management folks in Florida, try to manage the light as best we can, but it's still light. And coastal areas that might be, kilometers from the beach are also growing, and they produce sky glow, which is an indirect source of light, which we're discovering now is becoming more and more important in terms of affecting hatchling orientation. And also, you mentioned LEDs. That's an important aspect. So in the before time, before LEDs, the way to achieve energy conservation with lights is to use fewer lights and longer wavelength sources like high pressure sodium vapor, low pressure sodium vapor. Now with LEDs, we have cheap broad spectrum light, and people like broad spectrum light because they can see colors and it just makes them feel happy, I suppose. And so a lot of roadways and cities that were once lit with fewer fewer long wavelength sources are now being lit with more broad wavelength white sources. And so that sky glow I was talking about is becoming whiter and whiter, including more and more short wavelengths, and becoming more and more attractive to hatchlings near beaches. So, I mean, I see the challenges for light management as those two superimposed forces. One, more bad lights being visible from beaches, but also continued vigilance from people monitoring beaches, and then management of light as best we can to try to minimize and mitigate those effects from lighting. And the trend is hard to detect. Like, is it getting way worse or is it getting a little bit better? Maybe it's just staying the same. It's really hard to know because the number of hatchlings that die from lights is probably in the hundreds of thousands a year, but it's hard to track those trends over time, either annually or spatially. But we think we know what the right management strategy is, and agencies in Florida are working hard at that. And it's a never-ending challenge.
Right. There was a large review I read a while back. I think that you were a co-author on that had a lot of in the appendix. There were a lot of recommendations for types of lights and if they were conducive for sea turtles or not. Is that still in date? Would it be useful for us to link that in the description of this for anyone that's interested in sort of analyzing what kind of lights they should use?
Yeah. So in one of the publications I was on, we had an appendix with kind of recommended light sources, and of course, the light source that someone might want to use differs depending on what they want to do with it. Do they want to light up a pathway? Do they want to light up a balcony? And the most important balance, I think, between protecting sea turtles and having light that is still useful is finding that intersection point. So light that is still useful, that makes people happy, but doesn't affect sea turtles. And so these recommended light sources are a compromise between what sea turtles might want, which is a completely dark beach and no light at all, and what people living near a coastline might want. And that is at least some light so you don't trip in the dark and you feel safe. And you're not living in complete darkness. So there's a pretty good set of compromise light sources in that Understanding and Assessing Light Pollution document has, but also I might steer people toward a really great nonprofit group called the International Dark Sky Association. They focus on all the aspects of light pollution. So IDA, they focus on a lot of the wildlife aspects of light pollution, but also the human aspects of light pollution. So before we touch a little bit about what the incentives are for anybody to care about anything or do anything about anything, many of us have selfish interests. Well, there are selfish interests to tackle the problem of light pollution because light is not just harmful for wildlife, it's harmful to humans as well. It interrupts our sleep patterns. It even causes cancer. Like objectively, light causes cancer. Anyone who would like to, challenge that hypothesis and look at the relevant data, have at it. Just Google cancer and light pollution, and you'll find some interesting discussions and some data in support of the hypothesis. Light causes glare. It's not very efficient in terms of what we really want the light to do, like see bad people in the dark or find our way on a path. There are a lot of selfish reasons why humans should be dealing with the problem of light pollution that are beyond the altruistic reason of preserving in the environment and protecting habitat for charismatic animals that we like to have around.
Right. And I guess the last sort of piece with conservation is the technology and the development now. And you mentioned earlier the use of sort of something I don't know exactly what you're referring to, but like a LiDAR related with hatchling sea turtle tracking, and even like recently the use of dogs on sea turtle beaches and that sort of thing, and increasing efficiency for detection. But what are the like advancements in technology that you're most excited for and how are they going to be applied to sea turtle conservation?
The question of using dogs to locate sea turtle nests was a fun one that we explored. As it turns out, dogs are pretty good at finding things with their nose, and yes, indeed, they can find buried sea turtle nests, eggs that are, like 3/4 of a meter under the sand. They're pretty good at finding those, so they can enhance the detection probability of egg finding in nests on a nesting beach. But they're also very interesting tools for outreach. So a lot of people might have only a modest interest in turtles or the natural world, but are crazy about, like, quote, animals and love dogs. And nothing really opens up the emotion of somebody more than a cute little floppy-eared beagle, which is one of the dogs that we used for nest detection. And you just see people, light up. And then once they're sort of emotionally exposed, you can give them all kinds of conservation messages. And we found that it was really helpful. And so I kind of think that the idea of detection dogs used in sea turtle nesting beach work is really most effective if you couple both of those aspects, the scientific aspects of increased detection and all of the conservation outreach possibilities that open up with dogs. But you asked about what technologies I'm excited about. So our Inwater Research group is newly exploring the use of sonar cameras to detect turtles in the water. Detecting turtles in the water has been a problem since we saw a turtle head and then it disappeared, and we wanted to know, all right, what's it doing? Where's it going? How many are there? Waters are murky. You can only see so far. Even if the water's clear, sometimes it's choppy and you just can't see very well. We're using a sonar camera that essentially sends out multiple sonar beams and develops a video image of animals and structure in the water. And we're trying to use machine learning to recognize. Sometimes it's extremely clear. You look at the video image and you say, that's a turtle and it's a loggerhead. Even though it's nighttime, even though the water is incredibly murky, you can see several meters away, and you can count up turtles and identify them. Other times it's a little bit more smudgy, so we're investigating machine learning for identifying the smudges. We'd like to be able to teach the algorithms that, we verify that this smudge is a loggerhead or a green turtle, and it's this size or that size. And so we're hoping that this kind of technology sort of opens up an additional sensory modality to detect and count and understand turtles in the water, when before it was just a big opaque surface that was hard to penetrate.
That's a fascinating application of machine learning. That's something that there's so much untapped potential there for conservation work and turtle work in general. I'm curious with respect to your experience and touch and then wrap things up. But in the process of, what was the most interesting thing that you learned that you didn't expect in the process of writing your book, Our Sea Turtles?
Well, I wrote the book with my wife, Dawn, who is a wildlife artist and graphic designer. So I learned that it was fantastic working with her. We work together well. She's my wife, of course. You might expect that I'm fond of her, but she's also a fantastic collaborative companion. So we fit together well, yin and yang. I'm the pointy-headed science nerd, and she is the big picture aesthetics, common sense person. So both in the way we presented things in the book and in how I write about things, she lent a lot of editorial advice on how to broaden things up so that I wasn't turning people off with too much, inside baseball on the biology of turtles. Because the theme of the book is our sea turtles. So the idea is that, yes, these are animals. They exist in nature. They live fascinating lives. But fundamentally, we exist with them. We have effects on them. And we also benefit from them. We benefit from them in ways that we don't completely understand through, being connected in the same ecosystems like marine habitats. but we also benefit, emotionally. They're inspirational. I mean, a kid who sees a turtle nesting on a beach might get inspired and not be a sea turtle biologist, but be a scientist and start asking questions about the natural world, or at least have enough interest in nature so that they are an informed citizen, and they care about our links to the natural world. They understand them a little bit. And so we really wanted to present sea turtles on a slightly less academic level than a lot of books. We wanted to present concepts in fairly common sense form and throw in some charismatic images that help portray these animals, because I think that turtles are awesome, and as a group, sea turtles are awesome. People should appreciate that, and sea turtles need that. Sea turtles and wildlife in general need that broader human constituency, because without that, conservation is too great a challenge. And all of us as conservationists need to talk not just to each other. We need to talk out to all the people that matter. So we hope to do a little bit of that in the book.
Yeah, I mean, you mentioned like young people getting inspired by it. I remember distinctly that I think it was 6th or 7th grade, I would read our sea turtles in the back of the back seat of my mom's car going to school, and just it was, as someone, a younger person interested in it, it does have a lot of information, but it's conveyed in kind of beautifully illustrated and that sort of thing in an engaging way. And it definitely inspired me. I'm sure a lot of others. So it's a really cool piece. And for anyone listening, definitely a great addition to a library. So sort of round things out, I guess, the broader picture here of why should people care about sea turtles?
Why should people care about sea turtles? That's one of the questions that I really hate, but I like to answer, because it's a question that we need to be able to answer, and it implies other questions about like, why should we care about anything other than ourselves? It implies, what's in it for me? So you could say that about turtles or wildlife in general, like, what is it good for? Why should I care about them? It implies our, fundamental selfish sense that we want to know, what's in it for us? Why should we care about these animals that are, out there living their lives? Now, if you like turtles and you think they're cool, then the answer is obvious. They're charismatic elements of our environment. The world is a better place with them. They're fascinating. They teach us about the broader world. And it's obvious. It's the other folks that really need the answer to the question, like, what's in it for me? And I think there are two answers to the question. One, of course, is that everyone has selfish interests in protecting wildlife and turtles and sea turtles, because we're connected to them. All of these animals are connected to us through the ecosystems that they inhabit. Sometimes we have direct connections, sometimes we have indirect connections. Sometimes the things that affect these animals also affect us. So in saving them, we're also saving ourselves. There's some very real ecological reasons why especially certain wildlife species should be protected, and we should do everything we can to minimize harm to them. But even for someone who doesn't buy that argument, there's another aspect to the answer to the question, and that is that you might not care about turtles, but you should understand that a lot of people do, and there's a wide constituency of people out there that you should respect. And I'll use the example of So we don't happen to have any kids. But if a local school board wants to build a school in our neighborhood, I would support it. And the reason is, I mean, my kids aren't going to go to the school, but I'm perfectly willing to support it because I understand that educated kids are important for our community, and I'm part of our community, and I want kids that are educated with proper resources and good schooling to be part of my community and to help support the wider economic system that I trust I'll have. So things that aren't very specifically and directly good for you aren't the only things that we should care about. I mean, we should care about other people, people who care about the charisma of animals, people who care about science and conservation, and also realize that we're all part of this larger system that includes ecosystems shared with many other organisms, including turtles.
Well, that's I mean, really a testament to the fact that I think when you ask this question to a lot of people, there's sort of a go-to response, but that's a really just demonstrates the level of thought you've given to that and the complexity of and a lot of sort of appeal to the complexity of the situation of why should we care in general is a really good way to look at it, and also a bit of a stressful way to look at it because it's a philosophical question as much as a sort of question of biology. Out of all the places you've worked with sea turtles, where's been the most interesting for you to work or your favorite?
Well, I did some work in the Middle East, in the country of Oman, at some really important sea turtle nesting beaches. One for green turtles at Ras Al Had, which is at the very tip of the Arabian Peninsula. And then a very important loggerhead nesting beach on Masira. And it was very interesting because having come from Florida, which is also a very important loggerhead nesting beach, it was nice to compare and contrast the two areas, widely separated on the globe, but similar in terms of how those beaches supported loggerheads. And in fact, between the island of Masira and Florida, I mean, that's about, 3/4 of the world's loggerhead sea turtles right there.
Wow.
So it was extremely interesting to work on those nesting beaches and see the similarities in sea turtle natural history in those spots, and to understand population trends and threats. But it was also great to work with the people there because one of the sort of the universal aspects of sea turtle biology is that it thoroughly depends on sea turtle people. So we have an international sea turtle symposium. People from all over the world come, and they depend on networks of people for their science and for their management and conservation. There's a whole lot of citizen science out there in terms of sea turtles, and it's great to see how all of that takes place in different cultures. And the Arabian culture is quite different from U.S. culture, but they're fundamental problems. They're dealing with light management, just like we are here in Florida. They're dealing with fisheries. They're trying to interact with people who are catching fish and teaching them that there are these ways that you can catch fish in your nets that won't kill as many sea turtles. There are ways to have lights on your coastal property that won't disorient as many hatchlings. So there's still a lot of fundamental aspects of human behavior, sea turtle conservation challenges, and biology that are universal. And when you go to other areas, it's really nice to have that point driven home.
Right. And the Masira population is not doing so well, as I understand it.
It is not. In fact, I mentioned the Florida population is sort of bumping around and being static. So in the years that we've been monitoring that closely since the late 1980s, the Masira population has declined by about 70%. And whereas the Masira population was the world's largest population of loggerhead sea turtles, now it is not. But it's not because we've made a lot of important gains here in Florida. It's because the Masira population has declined drastically, unfortunately. And there are a few smoking guns to point to, like a shark gill netting fishery that takes place right off the nesting beaches during the nesting season. In one of the worst mortality threats you could think of. And it's so coming to grips with how to manage that fishery is a really important element to saving that population.
Yeah, so it's sort of a bittersweet thing that the U.S. has claimed to the title of most loggerheads, because it really historically would not have been the case, it seems like. But yeah, that sort of thing is a fascinating subject. I spent some time in Madagascar working with tortoises and just thinking about the issues with tortoise conservation there and hunting and that sort of thing. You have to think of it, the people too, the livelihood aspect of things it's different than the United States. So it's a complicated issue because we want to do the best for the turtles, but the people also factor into it. And in order to do good for the turtles, you have to have the people on board. So
yeah, humans are the most important element in conservation. And anyone interested in conservation should know something about like human behavior. There's a whole field in human behavior change because ultimately you want to get people to do something a little bit different so that they're not harming the resource. And humans are both rational and irrational. We're rational in the sense that show me the incentive and I'll show you the outcome. So we will often address our own best interests, but we're also irrational. We'll make quick decisions that are based on our tribal instincts from when we were sitting around a fire. And so we have a whole lot of cognitive biases that mean that we don't always make logical decisions, but understanding that helps us communicate and then achieve human behavior change. And they're experts in that field. And there's also a wide literature. So I would encourage anyone who's interested in conservation in general to every once in a while expand your view outside the biology, the animal you want to conserve, and think about what makes humans tick, because that's the most important element in conservation.
Right. Well, as our last question for folks out there looking to make turtle work a career, what's some advice or one piece of critical advice you'd have?
I would say if you want to work in the field of wildlife or want to study turtles, get out there, meet people, go to meetings, volunteer, be an intern. Eventually it's nice to go to a good program. Often graduate school is required, but there's nothing that is a substitute for meeting the people who will one day hire you, or introduce you to the person who will hire you, or introduce you to the agency that will hire you, or expose you to the really cool science that is going to be your gateway to a career of research. I guess in short, I would say get out there, meet people, do stuff, talk to people, and become known and know people. That's the most important thing.
Right. Throw yourself in the mix and don't say no to an opportunity. Right.
Yeah. And a lot of us are introverted. So we might focus on studying an animal that is itself kind of introverted because it satisfies our scientific sense to completely focus on this and then shut out the outside world. But I would encourage everyone to force yourself, go up and meet people, tell them about yourself, tell them about your ideas, comment on the paper they gave, and just, be at least facultatively extroverted to, go to meetings and try to put yourself out there.
Yeah, that's an interesting piece of advice I don't think we've heard before on here. So it definitely is struggle sometimes of personality types that can sit there for a long time behind a computer and analyze and look at things and do field work. It can be kind of, you get boxed in, but you have to
Yeah.
realize that there's no reason to contract down like that. Well,
Yeah.
I mean, this has been a really fruitful discussion. It's gone a good duration, but it's just been a really interesting in so many ways. And um, Jack had to leave. He has to go trap some turtles for a project he's working on. But we like to finish off with just a quick round of turtle trivia. So I don't, if you have to go, I totally get it. But I don't know if you have a random specificities encouraged turtle fact that you want to quiz me with. See if I can answer it.
Uh, all right. I don't know. We talked a little bit about turtles eating. You might be familiar with the esophageal papillae that sea turtles have.
Right. Yep.
What's the conventional wisdom as to what those are all about?
Well, yeah, that's an interesting question. I've heard that I mean, in leatherbacks, it's really useful for keeping down aqueous food items like jellies and that kind of thing. And when you're ingesting a lot of water, I can see where that could be something useful, is if you can get it down on a surface, because it won't just come back up, and keeping food items suspended in the esophagus. And I mean, that would be what I would say for that, but maybe there's some more nuance to it.
Yeah. So I mean, that is the conventional wisdom, exactly what you were talking about. But I mean, the broader aspect to it seems to be based on the expense of ingesting salt water. So it's not just water; it's salt water. I mean,
Right.
so marine turtles have this, but not freshwater turtles. And leatherbacks have it in spades. I mean, not
Right.
the papillae kind of line the whole mouth cavity and then all the way down the throat. So the whole eating slippery stuff seems to be supported by that, and the aspects of the physiological costs of ingesting salt water also seem to have something to do with it. So the esophagus is also pretty muscular, which allows the animal to like almost wring out the food before it swallows it. So it's ingesting as little of that salt water as possible. Otherwise, it has to deal with it uh, and excrete the salt. Sea turtles have these salt glands, and they're constantly excreting these extremely viscous, salty tears to get rid of salt.
That's
But but
interesting.
that but that's one of those cool things that, so a lot of that is supported by data, but in the end, it's kind of storytelling. it it's hard to know.
Yeah, right. I can imagine, how would you actually figure out, how much? Yeah, the function of it is sort of a dehydration mechanism is interesting, and that kind of makes sense. Is, yeah, it's
Uh,
contorting and moving like that. There you go. There's another study idea for a future student is
yeah,
figuring out
absolutely. Yeah, for any
how do you
students,
measure.
I mean, there's a thesis around every corner. Uh, yeah, I sometimes get approached by students who want to know what kind of unanswered questions are there. Oh my God, where do I start? There's just so many unanswered questions, and a lot of them are really important from an applied aspect, from a conservation aspect, but also a lot of really cool biological questions, and a lot of questions that span really cool biology questions that can apply in the real world to conservation as well.
Well, awesome. I think that's a cool note to end on, and this has really been a pleasure and an honor to sit down with you, Dr. Witherington. Thanks for joining us today. If there's anything else you want to end off with, I'll open the floor to you. If not, we'll call it.
I don't think so. I thank you very much for the opportunity. It was an honor for me as well. really insightful questions and a fun conversation. And I hope I use the opportunity to deliver some of my pet peeves and pet ideas and philosophical discussions in the conversation. So I think you gave me lots of opportunity. It's been a fun conversation.
Well, awesome. Thanks. And for all the listeners, we'll see you on the next.